All right, I think it's ready for the other team.
All right. Hi, everybody. Like Carrie said, thanks so much for joining us. My name is Greg Capareso. I lead the QIIME2 project, and so have been working on this for about 15 years now, including QIIME2 and its predecessor, QIIME1. I've got a bunch of information up here on these slides. Probably the most important thing for you to grab right now is this slides link that I have here. It's a little bit long. I was going for a short URL, but it got pretty long. So just go ahead and type that in. That will let you bring up this slide deck. You could also scan the QR code. That'll bring you to these slides. And then you can just have it in front of you. There's a lot of links throughout these. There's a book, a Jupyter book that we're going to be using throughout the course of the workshop. And so that is linked from the bottom of this. But once you get these slides up, then you should be all set. Does anybody need another minute to get these up? Just raise your hand if so, and I'll. Oh, okay. Yes, and they are linked from the agenda, which you have. Or if you just click on the individual sections in there, it'll bring you. Oh, okay. We'll. Got it. So if you didn't hear that, if you click in the agenda, just scroll back to the beginning of the slide deck and you'll be on this slide. Okay, anybody need another minute? Okay, great. Okay, so I just want to continue on with the introductions a little bit. So we just heard from Carrie. I'm Greg. I've got a few folks here from my team. And so in the back, I'll just have people wave or stand up for a second. We have Chloe, Colin Brislon. Colin's not on my team, but joining us as a TA. Hannah and Colin. And then we just heard from Carrie, and then Candace and Kate. And so we've got a big instruction team here. The purpose really is so that you can hear from a bunch of different voices. And as people get stuck throughout, you can raise your hand if we're working on something hands-on, and somebody will run over and help you. And so we're really here to support you in learning these tools this week. And this slide is here just, so you can put names to faces if you want to like grab somebody, or we're having a conversation with one of the instructors and want to flag them down or look for them online. This is everybody. I also want to just start by acknowledging our funding sources. Carrie's team and my team are both funded through the NCI Informatics Technology for Cancer Research program. They are the main funders of this, and then we've got some others in there that are supporting aspects of this. And so today what we're going to be doing is talking about some very introductory microbiome analysis. And so I'm going to introduce the QIIME2 platform, just sort of a very high level view of it and what it is and what it's become, and some of the features that people find very interesting and useful about it. And then we're going to be talking a bit about reproducibility in bioinformatics. We're going to be talking about cloud computing. So a big focus of this is on resources that you can use for running your QIIME2 analysis. Then we'll have a lunch. And then in the afternoon, we're going to be doing some hands-on work with Galaxy to use QIIME2. And then a couple of activities in there. Tomorrow, we'll be switching gears and we'll be using Docker to run analyses on your computers. I'm going to talk about this a little bit more later. And then Friday, we're going to be doing some more hands-on work with a relatively new addition to sort of the sphere of QIIME2 plugins. one that's called Q2FMT for fecal microbiota transplant. This is a plugin that's being developed by Chloe, who's here with us this week as part of her dissertation work. And this is intended to really give you an introduction to just some of the diverse things that you can be doing with QIIME2, how you can learn about other plugins that might not be included with sort of the core distributions of QIIME2 that folks are working with. Okay, so the next thing I want to mention is that I mentioned that my group leads the development of QIIME2. But this is really a sort of a global network of collaborators who are working on this project. This is well illustrated by the author list on the original QIIME2 paper. We had over 100 co-authors there. I think we had over 50 different institutions represented and over 10 different countries represented in that list. And so while my group leads it, there's folks all around the world who are working on it. And those folks are either contributing plugins, so they're building new functionality for you to use with QIIME2. They're providing technical support on the QIIME2 forum. They're writing documentation. There's a lot of ways that folks contribute to the ecosystem. And it's been very widely used. So at this point, QIIME1 and 2 have been cited nearly, or actually just over 50,000 times. Frequently referenced in patent applications and used in clinical trials. And we've had stable funding for the project for almost 10 years now. And so with some of our most recent funding, what we've been doing is expanding into new domains of microbiome research. And so where we really started as an amplicon analysis platform, we are now supporting metagenomic data analysis. We are working with some folks on supporting metabolomics analysis, proteomics analysis. And the goal here, all of these sort of with the microbiome perspective, and then the goal is to build this integrated microbiome multiomics data science platform. And so that's kind of where we're going. And I like to throw these numbers up here, not to boast about the number of citations or anything like that, but just to show you that like this is a very widely used, very trusted tool in the field. And so, yeah, like I just mentioned, the goal is while the initial focus was on amplicon analysis, which gives us information about taxonomy, where we're going is supporting these different layers of microbiome analysis. And what we hope to do is integrate those layers to learn something about the functioning of these systems and how they interact with their hosts or how they can be used to advance biotechnological goals. So all of that said, what is QIIME2? So a lot of folks think of it as this amplicon analysis tool. It definitely is that, and that's what we're going to be focused on this week as we're learning. And so the idea here is that QIIME2 will take you from raw sequencing data as it comes off the instrument through steps like demultiplexing, quality control, developing what we call the feature table. And so the feature table will be essentially can be different feature types, but most typically it might be what's known as an amplicon sequence variant or a unique amplicon sequence that you observed in your analysis, and counts of those on a per sample basis. And that becomes the core for a lot of the downstream analyses that we do. And we focus a lot on that. We're going to focus on that table a lot in this workshop because it's a very generally applicable data structure, data type in bioinformatics. And so if you think about an amplicon survey, that might be amplicon sequence variants on a per sample basis. If you're doing a metagenomic survey, that could be something like bacterial species on a per sample basis. It could be something like functional genes or pathways on a per sample basis. If we're moving into proteomics or metabolomics, that could be proteins or metabolites on a per sample basis. And so a lot of the analyses that we do sort of converge on that feature table. And regardless of what you're doing upstream, so QC is going to be different, obviously, whether you're working with a sequencer or a mass spectrometer. But when you get to that feature table, a lot of the analyses that you want to do become very similar to one another. So a lot of the analyses that we're going to do this week are things that you could just as easily be doing on metabolomics data or metaproteomics data or metagenomics data. And so while we do have our amplicon analysis workflows, QIIME has been used in other domains. And so it's been used in the early days of the pandemic. We used it for sampling SARS-CoV-2 genomes, for building phylogenies, for studying how the virus was moving around the world, and for us, how it was being introduced into Arizona. I mentioned that that we now have this metagenomics distribution. We consider this to be in a sort of alpha or testing release stage, but this is something that you can go and install on your computer today if you want to. And the idea is that this can take you from reads, so sequence reads through your downstream analyses. And it can do things like assemble reads into contigs. It can do mag calling. And generally what we're doing is we're interacting with other tools that support this. And a lot of what we do with QIIME2 is focused on the connections between these tools. So making it so that you can easily swap out, for example, a tool for assembling reads into contigs. And that facilitates, first of all, the use, because if you learn how to use QIIME2 on amplicon analysis, the way that you're going to interact with it is essentially identical to how you're going to use it for metagenomics analysis. The tools you use might be different, but the way that you interact with it is going to be the same. And so similarly, by having different tools that can support similar functions, and then us mapping, say, like similar data types onto each other behind the scenes without you having to worry about it, it facilitates doing things like comparisons across different methods that you might want to use. And so do my results differ if I, assemble contigs with tool A versus tool B? Become very straightforward questions to answer in the QIIME2 ecosystem. And so a pipeline, I think of it like an assembly line. Like there's this one thing, you put some input in, you get some output out. With QIIME2, what QIIME2 essentially at its core has become is a framework for building these pipelines or workflows. It's still very much focused around microbiome research, but it's become much more generally applicable. And I'm going to talk now about some of the things that have driven this, like why people are using this in different domains. And then this is intended to serve as an introduction to how we're going to be using QIIME2 over the next few days. And so, again, we support extensive amplicon analysis. This is what we're going to be focused on. Sorry, I seem to have some duplicate slides in there. Okay, yeah, so some of the things that make this most interesting for folks is, number one, our ability to create very portable and very portable interactive visualizations. And so, for example, this is one of our newer interactive visualizations. On day 3 of this event, we're going to have you generate plots like these. And what this is doing is it's tracking what we would refer to as microbiome engraftment following a fecal microbiota transplant. And just to illustrate that these are portable, like if you were to scan that QR code, you should be able to pull this up on your phone and interact with it on your phone. This is really handy for example, for like presenting posters or giving talks like this. Becomes very easy to allow the folks who you're communicating with to interact with your data, even if they don't have QIIME2 installed on their computer. Another thing that has made the platform very popular is the ability to work with QIIME2 through different interface types. And so the way that I think about this is that, if we think that researchers sort of generally are spread along this continuum of how sophisticated they are or how advanced their computer skills are. What we do with QIIME2 is we make it so that it's accessible to people at different stages who are at like different points on this spectrum of computational skills. And you can do all of the same things regardless of which of these interfaces you're using. And so, for example, it's not like you can do more or better things if you're working, with the QIIME2 API, like in a Jupyter notebook. You can do all of the same stuff on Galaxy. You just select the interface that you're going to be most efficient with. And so, the data science folks tend to gravitate toward using the Python API. Most users out there kind of fall in this, like what I describe as power users here. This is going to be folks who are using QIIME2 through the command line. And then sort of what I think of as more like the domain scientists, so biologists or clinicians who want to analyze their own data, but don't necessarily have the computational background to be using the command line or using the API, can interact with QIIME2 through the Galaxy interface. And so the Galaxy interface, this is what we're going to be focused on today, is a fully browser-based approach for interacting with bioinformatics tools. The Galaxy team, like Carrie mentioned, is also funded in part through the same program. And so we've worked with them to make QIIME2 accessible through Galaxy. We're going to be working on a server called cancer.usegalaxy.org. And if you want to learn more about this, we have a full course on our YouTube channel. It's actually from a 5-day, 7-hour day workshop that we did. And it basically goes into this data set that we're going to be working with here in a lot more detail. And so if you want to learn this, this is a relatively short event, so we don't have time to go into as much detail on things like, the theory and approaches behind some of the diversity metrics that we use. That kind of stuff is covered in this online course. And that's all using Galaxy. Galaxy is wonderful. There are some limitations though, and I think you'll see those when we're all working with it this afternoon. When we're using this particular system, we're all going to be using the shared computer resource that is provided by somebody, far away on their computer hardware. There's actual, machines doing work when we're clicking jobs. And what that means is that we can end up waiting in a queue. So there may be other people whose jobs are queued up before ours. And so that can take some time. We're working on some approaches for distributing Galaxy with QIIME2 so that you could run this on your own computer. Or if you have, say a local installation of Galaxy, like if your high performance computing group runs a Galaxy server, you can ask them to install these tools in there. But the alternative, of course, is to use your own hardware. And so tomorrow, so Wednesday and Thursday of this week, we're going to be focused on utilizing your own computational resources to run these tools. And to do that, we're going to be working in a containerized environment. And so that is essentially a virtual machine. We're going to be hearing from the ITN team today just to learn a little bit more about those, to contextualize them and learn how to use them a little bit. But basically what this is this little virtual machine that you're going to be running on your own computers that will provide a consistent environment across everybody's computer to use QIIME2. Both of these approaches are things that you can take away with you after this course. And so this cancer.usegalaxy.org server is publicly accessible. You can get accounts for free on there. And I have had clarification from them. It doesn't need to be for cancer research. So you can use this for whatever it is that you're working on. And then this containerized environment is something that you can run on your own laptops. These containers that we're working with are also very portable. That's kind of the purpose of them. And so one really neat thing about this is that if you're running this, say, on your own computer, as you're learning QIIME2, and you know are working with some of our toy small data sets that we provide, at some point you're going to be ready to transition to your own data, and your resource requirements may exceed what you have in your laptop or what you have access to through your institution. These containers are very easy to transition over to cloud-based servers. And so you could then be interacting in this exact same way, but on some platform where you are paying for some time to use cloud computing resources. And so we purposefully picked these two ways of using QIIME2 because they allow us to give you something that you can walk away with and run on your own. Now, while I'm talking about this, we have provided some information before the course on getting Docker installed on your computer. I'm going to show you a link a little bit later on that has instructions for loading up the container that we're going to be using in the workshop tomorrow. It would be ideal if folks, when they go home today or go back to their office today, if you could run the steps to just start the container up on your computer. If you do it here and everybody's trying to do it at the same time over the same Wi-Fi network, we may slow things down because it's going to be downloading. It's not a huge file, but I think it's about a half a gigabyte. And if we all hit the Wi-Fi to try and download that at the same time, I'm just not sure what's going to happen. And so we will provide you the link that we'd like you to follow to go download this. There's also two options there. They're Docker and Podman, and I have both of these links up here. So Docker is probably the one I would assume most people have heard of Docker. Podman is an open source implementation of that's distributed by the Red Hat group. And so it offers some benefits that Docker doesn't have. And so Docker can be hard to get installed on institutionally owned computers that have restrictions on them. And so it requires administrator access, and a lot of institutions won't allow that. Podman is an alternative that does not have that. And it's basically just like, for the purposes that we're using it for, it's essentially a drop-in replacement. And so when you would run a command that starts with Docker, you run that same command, but swap out Podman for it. And so we've got, again, I have a document that we're going to be pointing you at that provides all of these instructions. Okay, so the other thing that I wanted to talk about right now is how QIIME2 facilitates reproducibility in bioinformatics. And this is, of course, important for future consumers of your research work who may want to know what you did, how you did it, in order to say, like, validate what you did, make sure that they could reproduce it on their own data. But there's also some huge benefits that you directly can realize from working with tools that better support reproducibility. Two examples of that are when you come back to an analysis several months after you ran it. And so imagine you have some situation where you run an analysis, you submit a publication or you submit your dissertation, and then a couple months later, somebody comes back, say, or a reviewer of that manuscript, and they're asking for some very detailed information about what you did. And so, for example, what version of the NumPy library was installed on the computer where you ran this because there was a bug that may be impacting the results that you're seeing. A lot of us don't have that written down. There's just an enormous amount of information like that that is hard for humans to record. And so QIIME2 behind the scenes records all of that information for you. And we're going to show you some really neat examples of this at the end of the workshop. And so when we get to the end of the workshop, we're going to go back and we're going to look at everything we did over the course of the last few days. And you can then take those resources home with you just as your notes from this event on all the commands that you ran. The other way that this benefits you as a user directly is when you come to us for technical support. And so we run the QIIME2 forum, and this is really like your main resource for getting technical support on QIIME2. Also a very useful resource just for discussing microbiome analysis in general, for learning about new tools, for finding new resources. And so a lot of times, we'll get a tech support question from users, and we'll be trying to figure out, okay, what did they do at this step? And sometimes that can be hard to communicate just because, we have different languages to talk about these things. And so the way that, like we technically think about something may be different than how you're thinking about it. So it can be hard for us to narrow in and understand the question, understand what you did. Often what we'll do is we'll ask you to share one of your QIIME2 output files with us. And then what we can do is we can view the data provenance. We can see everything that you did to generate that result. And that really facilitates us providing technical support. Back in the QIIME1 days, we did not have anything like that. And this has saved us a huge amount of back and forth. And so what that means for you is that you can get the answer to your question more quickly and move on with the stuff that you're actually interested in, rather than going back and forth with us, trying to debug what went on with some workflow. And so we're going to talk on later in the workshop about a tool that we have called Provenance Replay. But essentially the information that QIIME2 is recording is detailed enough that we can take that and we can generate new executable code. So we can say generate a Python script or a command line script from any of the results that you have created. And so the data is not necessarily always represented in the provenance, but this is like how you got there. And so we will come back to that a little bit later in the event. The other thing I'll mention about QIIME2 and really like what has enabled us to expand into these different domains is that QIIME2 is built on a plugin architecture. And so all of the actual bioinformatics analytic functionality in QIIME2 comes in the form of plugins to the system. And that includes the stuff that people think of as like the core QIIME2 functionality, all is in the form of plugins. And so what this enables us to do is work with developers all around the world to build plugins that can then be used in the QIIME2 ecosystem. And so, for example, my group, our expertise is in the bioinformatics and software engineering associated with amplicon analysis, a little bit on shotgun metagenomics analysis. We're not experts, for example, in metabolomics analysis or metaproteomics analysis. So what we do is we end up working with developers out there in the world who are experts in those fields and who are interested in making their tools accessible through QIIME2. The things that attract those developers are the things that I just mentioned to you. And so when a developer writes a plugin, they don't have to think about adding Galaxy support and adding a Python API and adding a command line interface. That's what the QIIME2 framework takes care of. And so they can focus on implementing their bioinformatics methods, and then they get the benefits of this data provenance tracking and these different interfaces by virtue of making their tool available through QIIME2. We have a relatively new plugin developer manual. This would be more for folks on the data science end of the spectrum, folks who are interested in building and distributing bioinformatics tools. We also spend a lot of time doing workshops. That should be obvious since you're all here at a workshop right now. But we have done on QIIME and QIIME2 well over 100 workshops at this point, all around the world on 6 continents. And so we've done some of these online. You can find those like during the pandemic. Obviously, we transitioned to doing a lot of this online. And we hosted 2 workshops that we turned into full YouTube courses on there. And so one uses the command line, one uses Galaxy. They cover a lot of similar content, so you could use one or the other of those. And I mentioned the YouTube channel a few times, but this is where you can go to find all of this content, and we do have them organized into courses. I also mentioned the QIIME2 forum. This is the hub of the QIIME2 community. If you don't have an account there already, I recommend just go and create one. You can also read for free though, and we have I believe, I don't have the number up here, but we have like on the order of 100,000 page views per week. About 20% of those are from people who are logged in on the forum. And so we know that there's a lot of people who are active, but there are also a lot of people who simply use this as a resource for learning. And so there's been so many questions answered on there that chances are if you just go to the QIIME2 forum, put a few words in on what you're searching, what you're having trouble with, or what you're wondering about, the question has probably been asked for. Now, one of the things that has made this forum successful is that we very actively moderate the forum. And so several of the folks in this room, Colin Brislon, Colin Wood, Chloe, Hannah, are active on the QIIME2 forum just about every day. And so if you've asked questions on there, there's a good chance that one of these folks who's in the room right now has answered your question. Basically what we commit to is that if you ask a technical support question on there, we're going to respond to it within 2 working days. We do this all for free. And so there's no charge to you as a user to use this forum. And from my perspective, this is one of the things that has made the platform so popular, is that there is this way to get support for it from the community. This is also a great way to connect with people who have similar interests to you. And so Colin Brislon, who's sitting over here in the audience, and I have been interacting on the QIIME1 forum and the QIIME2 forum for probably 10 years now. And we just met in person yesterday for the first time. I also know of several people who have found postdocs via forum interactions. And so we have a free job board on there too. You're more than welcome to post to that job board. It's not super active, but every couple months there's a post going up on there. But just a free way to like get, say, your job ad in front of a bunch of people who are using QIIME2 on a day-to-day basis. Okay, so some of the stuff that's coming soon. So we are, like I mentioned, working on, well, we're sort of in discussions on a metabolomics distribution. Some colleagues of mine recently published a protocols article on how they are already doing metabolome analysis with QIIME2, and we're now working on that with them to figure out like how to fill some of the gaps. So how do we make it so that this is sort of like a platform that you can use for this? There's a lot of overlap there just because of the technologies used with metaproteomics research. And so those two are kind of evolving together. We also have new ways to interact with QIIME2 through R. We will point that out to you a little bit during the tutorial today, or tutorial this week. We're not going to get into that in this event, but if you're an R programmer and that's how you want to interact with software, we have some support for that. And then we are planning some additional support for that. I mentioned this updated developer documentation, and then we now what we're doing is we do 2 formal releases of QIIME2 every year. We do a release in April and we do a release in October. And then generally weekly, we're pushing new development releases out. And so that's like, you have two options there. Like if you want to use the most stable tested version, then you're looking for one of our release versions, either in October or an April release. If, on the other hand, you want access to like the latest cutting edge functionality that we're working on, then you probably want our weekly builds. And the weekly builds, they are very tested in terms of all of the analytic functionality. There may just be some rough edges at times because we're just pushing things in as we test them and as they're ready. But then when they get in people's hands, people find all kinds of creative ways to break them. The other thing that I don't have in here, which is very new and very exciting, is over the past year, we've completely reworked the parallel computing framework in QIIME2. And this was largely to support our transition into metagenomics analysis. And so there's new functionality in place that would allow you, for example, to distribute a job across, say, 20 different nodes on your university HPC cluster. If you need help with that, it's something that we can chat about. Probably the forum is the best place for us to communicate about that. But if you have questions, let me know this week. And one of the things that I'm hoping to get from all of you this week is your input on where we should be going. So what do you want to do with QIIME2 that you can't right now? And this type of feedback and these types of events have literally driven the development of the platform. And so when we were doing QIIME1 events, I would hear about things, I would see things that people were struggling with on the forum, and that led us to do a complete redesign of the system.